I'm Dr. Asemi, and I'm a board certified lab director in vitro fertility center. I'm going to talk about the IVF laboratories of the future. Okay, let's review what is the infertility and its treatment first. Inability to conceive and despite frequent and unprotected intercourse for at least a year is infertility. But if the couple are younger than 35 years old, this time is going to be less than 6 months. The globally 48.5 million couples experience infertility, and infertility affect 12 to 18% of couples in the United States of their reproductive age. Up to 15% of all infertility cases may remain unexplained. Okay, what are the treatments? The treatments are conventional IVF, which we add a sperm around the oocyte and check the day after to see that if they're fertilized. And then the rest is going to be same as the second treatment, which is injection of a sperm to the oocyte. I'm going to explain second one more in detail. And then the both treatment needs to have the process that is involved, monitoring and stimulating an individual ovulatory process and removing an ovum or ova, which we call it egg retrieval. And then we're going to have them culture for 6 or 7 days and check them. And after they become the blastocyst, or some center, they transfer the embryo at the cleavage stage, which I'm going to explain that more in detail, to the with the intention of establishing a successful pregnancy. Okay, let's review what happened in day of the egg retrieval. The egg retrieval first happens in the OR operation room, and after the embryologists get the oocyte, they're going to get prepared for sperm sorting. And after they have the good sperm, and then they're going to get prepared to do the microinjections or intracytoplasmic sperm injection. Then they're going to monitor the fertilized zygote to day 6 and 7. They become the embryo and blastocyst. Then based on the cycle plan for the patients, they're going to do the transfer or cryopreservations or biopsy to send the cells to the genetic companies. Okay, now we have oocyte, we need a sperm. For sorting a sperm, we have two methods, traditional method and then the modern method. The traditional method that is very regular to all the IVF centers in the United States and worldwide, which they use the culture media, they wash the sperm, and then they use a centrifuge to sort the motile sperm than the non-motile one. Okay, when they use the centrifuge, they're going to make the reactive oxygen species, which is not good for the sperm DNA. Then the second method, which is happened to be used in a modern IVF lab, which we call it microfluid sperm sorting. There is a slide and has a chip, and then we add the sperm. Then we're going to pick up the most motile sperm with a better DNA condition at the other side of the chip. And there are some articles that claim that we're going to get better sperm based on the DNA fragmentations and morphology of the sperm. Okay, now we have oocyte, we have a sperm ready. Now we need to do the microinjection. As you can see in this picture, embryologist needs to sort to select the very good sperm, which is going to be depends on sperm motility and sperm morphology second, but motility is more important. Then when they choose the sperm, they're going to go and inject this sperm to the oocyte. As you can see here in this picture, they're going to inject the sperm. This is a sperm, and they're going to inject the sperm into the oocyte, and this is the mature oocyte. Okay, this technique is very meticulous and needs a lot of training. The training takes sometimes 5 years for the embryologists become senior on this technique. Therefore, we cannot ask somebody that graduate from the school come and do this technique. Therefore, this is another thing that you explained about shortage of nursing. We have shortage of embryologists, and especially shortage of senior embryologists, because each embryologist needs at least 5 years after they are started to be the embryology to train for this procedure and the next procedure that I'm going to talk later. Therefore, there are, after this procedure, they just have other procedure based on the base procedure, just intracytoplasmic sperm injection. They have some other procedures that makes them to do this procedure easier and faster, because the time is very, very important when you work with the oocyte. Okay, the next one, it was called intracytoplasmic morphological selected sperm injections, which sometimes you heard as IMSI method. In this method, they use the microscope that make a sperm 7,000 times bigger. As you can see in this picture, compared to this picture, the sperm is so 7,000 times bigger, and then the embryologist can see the morphology of a sperm so much better, and then they can choose the most normal one to inject. And then the regular ICSI microscope, which is this one, it makes the sperm 400 times bigger. But the problem is this microscope is very, very expensive. Second, the problem is just less than 1% of the patient, there is a possibility that they don't have any normal sperm. Therefore, even this microscope going to be not useful. But because it's very, very expensive, usually just IVF lab, they prefer to have the regular just microscope to use for the ICSIs. But that could be the future of the IVF labs, that we're going to have this microscope so much cheaper than all the embryologists can use this for doing the ICSI. Okay, another variant of ICSI is PICSI, which we call it physiological ICSI, and the aim is to make the sperm selection less subjective and not dependent on the laboratory staff. As I just told you in previous slide, embryologist is the one that go and pick the best sperm. Embryology choose the sperm in the previous methods, but in this method, there is another very important thing that going to select the sperm for us, which is the binding the sperm to the hyaluronic acid. Okay, what is hyaluronic acid here? Because the egg and cumulus around the egg has the hyaluronic acid. Therefore, a sperm that who are the mature, then they're going to just bind to this cluster. Therefore, this dish that we're going to use to take the better sperm has the hyaluronic acid at the bottom, as you can see here. Then the sperm who has the better spermatogenesis and they are more mature going to stick to this dish, and then embryologist going to get just this one and then take them and then do the macro injection. It's very hard procedure. It takes a lot of time and a lot of training, and it's not really something that is going to happen in 1 hour. Sometimes it takes 2 and 3 hours to pick all of this sperm. Therefore, it needs a lot of training as well. Okay, what is this technique is good for? This technique is good for the patients who has a lot of DNA fragmentation in their sperm, and then the one that has an unknown origin of infertility, and the patient with the repeated miscarriage, which they think that is going to be because of the male factor, and then the previous IVF ICSI cycle failure anyway, going to be very successful with this method. Okay, Now we have a sperm, we have oocyte, we did a microinjection, we get the zygote, which is the fertilized egg. Therefore, we need to culture them to day 6 and 7. What we use for culturing them? We use the incubators. The incubators, there are three type of the incubator that has been used in the fertility centers. The oldest one is the box incubators, which is they put 2 or 3 patients in one box incubator, and then a benchtop incubator, which is, you can see the benchtop incubator here, then has a chamber for each patient, which we prefer that. But if the traditional IVF lab, they use the box incubator, whenever they take the patient's dish out, there is a possibility that they're going to jeopardize other patient dishes in this incubator by exposing them to the atmosphere, oxygen and carbon dioxide. I have to emphasize that carbon dioxide and oxygen in the incubator is supposed to be between 5% to 7%, based on the altitude of the lab, and the oxygen percentage is supposed to be 5% based on all of the literature that they see, whether embryo development by lower oxygen level. Therefore, those incubators are not really good for embryo development. But there are some fertility centers, unfortunately, they use them, but they get the very good embryo development anyway. But we recommend they use the benchtop and they get better embryo development. In our lab, we use another very advanced incubator, which they call it time lapse. Time lapse means that first of all, each patient going to have their own chamber, each embryo going to have their own place. And then, as you can see here it shows that each embryo going to have their own place. And then they take a picture of the embryo development. Every 5 minutes, they take a picture and make a video. Therefore, we don't need to take this embryo outside and check it under the microscope and expose them to the atmosphere. And this is very important. We're going to mimic the fallopian tube atmosphere here by just not taking this embryo out. Plus, we get a lot of information from these pictures that we call it morphokinetic parameters for the embryo development. And we can just study these morphokinetic parameters to see that which embryo is better, has a better development, then we can transfer that, or we can freeze that for the future transfer. Okay, I just mentioned here that morphokinetic a lot. What is morphokinetic? Morphokinetic refers to the time-specific morphological changes during embryo development, which providing dynamic information on a fertilized oocyte. And this detailed information is a morphological selection of embryo and blastocyst with high implantation potential to be used in infertility treatment. As I just told you, this is the differences between when we take the embryo out and check it under the microscope personally, than you use all of this artificial intelligence data. And therefore, based on the recommendation of American Association of Reproductive Medicine, we need to transfer one embryo for the patient who are younger than 35 years old. Therefore, by single embryo transfer, we can reduce the incidence of multiple pregnancy, because before we had a lot of multiple pregnancy. Nowadays, by this technique, we can have the better pregnancy rate by just single embryo transfer, and just reduce the incidence of multiple pregnancy for our patients. Okay. Let's review the different human embryo development before we watch the video from one of our patients' embryo development. Then you are familiar when you watch this video. Okay, the fertilization, it means that when 2 polynuclei, which one is from a sperm, the other one is from egg, just merge together, that is the sign of the fertilization. We have 2 polar bodies. This is the sign of fertilization that we are expecting after conventional IVF to see in IVF lab. Then we have cleavage stages, which the zygote divides into 2 cell, 4 cell, 8 cell to 16 cells. Then from this stage, we have morula, which is the ball of the cells formed after cleavage. And then this is a very important stage, therefore, because the embryo is going to switch to their own genetic component. And this is a very, very important stage for the embryo because they're not using their mother material anymore. And then at this stage, they're going to push these cells to two pools, which is one is going to be the inner cell mass, which this ball here is the inner cell mass, and the other side is going to be trophectoderm cells. Inner cell mass is going to make the baby, trophectoderm is going to make the placenta. Then we need to have a good chunk of the cells for the inner cell mass and good chunk of the cells for the trophectoderm to have the great rate in their pregnancy. Okay. Now I'm going to show you the time-lapse video from one of our patients, and you're going to see the embryo development here. You can see the two polynuclei here, that is the fertilized oocyte, and then you're going to see the cleavages, and then after a while, you're going to see the morula. And this is video from day 1 to day 6. Then that is a 6 days video. But then you're going to see the hours here. What are they? These are the hours that these embryos get to these developmental stages. And these hours are very important because if it's going to be faster or slower, it shows us that there is something not good. When we have a special hours that based on the study and artificial intelligence data, that we need to make sure that the embryo is going to be at that range. And as you can see in this blastocyst, there are some of these inner cell mass, they just pop out. And this is the hatch blastocyst. And this is a very good blastocyst. And for this patient, we did a biopsy and then sent these cells to the genetic company. And then if it became euplate, we transferred them. Okay. I just talk about the oocyte retrieval, and in oocyte retrieval, we get some mature egg and we get some immature eggs. Some of the immature egg are going to be very immature, which we call them germinal vesicle. This germinal vesicle, we cannot do anything right now in the lab with them. We cannot inject them, and even we keep them one day, they're not going to be good for the ICSI because there is a possibility that we see the nuclear maturation, but the cytoplasmic maturation is missing. Therefore, they cannot make a good blastocyst. And nowadays, there are some study goes on that they try to have some culture media to make this oocyte more mature for use in the lab in the same time. Because from the day retrieval and 2 ICSI, it's supposed to be 1 day. It's supposed to be 8 hours between them. We cannot keep the oocyte more than 8 hours, and then oocyte goes bad. Therefore, we need to have the system that they make this very immature oocyte to the mature oocyte in 8 hours to 10 hours. But right now, we are waiting for that, and all of the embryologists hope that this is going to happen very soon, because we get few, sometimes a lot of the immature oocyte that we cannot do anything with that. Okay, then another technique that right now is using in England and Welsh, but not all of the world, and even in United States is illegal, which they call it mitochondrial replacement. In this method, women with mitochondrial diseases help them to have a healthy genetically related children. As you can see in this picture here, the technique uses part of a donated oocyte to replace disease-causing faulty mitochondria, and the child would inherit the DNA from the third person. Another very interesting technique that is going to be the future of IVF centers and embryology lab is CRISPR-Cas9, which is the technology that may allow for genomic modification of a heritable disorder through the treatment of zygote or oocyte. And then I have to say that this is the legal in the United States, and many hospital and biotechnology companies, they're using CRISPR, and these as a trials right now is a clinical trials, and these trials are regulated by FDA, and we hope that in future the embryologists can do this CRISPR technology in the lab to modify some of these genome that is going to cause the heritable disorders. But it needs a lot of regulations worldwide and in the United States to make sure that it's going to be in a good hands. Okay. The next one is fertility preservations in females. Probably you heard around your friends and family members that they're cryopreserve their oocyte. Yes, we have the cryopreservation of the oocytes for the women who have the cancers before cancer treatments, and the one that wants to postpone their family making for the later age, then we're going to cryopreserve the oocyte between the age of 25 to 35, and we prefer to do that before the age 37. And then another preservations for female is going to be ovarian tissue cryopreservation, which happens for the teenagers who got the cancer. And then for this technique, again, we have some barriers. When we thaw these tissues, we need to have a better maturation of these follicles to oocyte, which is under the study. But because for those teenagers, we don't have any other way to do the cryopreservations for their fertility. Therefore, this is something that happens, and hope for the future that they're going to get the technique that we get these follicles more to the more mature oocyte. And the third one is the complete differentiation of the pluripotent stem cells into the gamete, and this is under study as well. But that is going to be the near future of the IVF centers and embryology lab, that each embryologist could do this technique and get a mature oocyte, hopefully, and mature spermatozoa as well. And then, as I just told you here, and this is, I'm going to emphasize that future of embryology lab is going to be about the stem cell biology. And then, therefore, injection of primordial germ cell derived from the induced pluripotent stem cells into the appropriate gonad to stimulate oogenesis or spermatogenesis and permit natural fertility, or even a standard IVF, going to be the thing in future. But is under the study, and lots of literatures and investigations going to be happen, and it happened in the animals, especially mouse. But there are some barriers for the humans that they now is under a study to take those barriers off. And then maturation of primordial germ cells derived from the induced pluripotent stem cell is another hope for the future, and these are very near future. But needs a lot of regulations as well for make sure that it's going to be happens in the right way that it's supposed to be. Okay, then another feature of the IVF lab is going to be genetic testing, which is right now has happened in most of the just IVF centers in the United States, as far as I know, and some worldwide, but mostly most of the United States fertility center, they do this type of the genetic test in 95%. And it's very common in the United States, and they do the trophectoderm biopsy, and then next generation sequencing, and the technique that they're going to test and evaluate the embryos for the extra or missing chromosomes, or for the monogenetic disorders, or even for the structural chromosomal rearrangement. There are two more techniques that is under the investigation, which called PGTP, to evaluate the embryos for greatest likelihood of resulting in the healthy offspring with reduction in disorders such as heart disease, cardiac arrest, and diabetes. And then the next one that's still under the investigation, which is they call it PIMS, which this technology let embryology to check for the DNA methylation in the embryos. Okay, when we talk about the future of IVF centers and then IVF lab, we have to talk about the artificial intelligence. We cannot do anything without artificial intelligence. And then we need to have a digitalized lab. For having the digitalized lab, we need to have a very strong software system that is going to support the lab. We're going to do the quality control, quality assurance, and quality management by using the probes and using the artificial intelligence and using the digital programming. In our lab right now in Invita Fertility Center, we are using all digitalized. We don't have any paperwork for the patients. We just use everything digital and in a real time. Like, for example, for quality control and quality assurance, we do the real time. It saves a lot of time for us. And with the shortage of embryologists and senior embryologists, unfortunately, we have no other choice to just save the time and just use our embryologists to do the most important things, which is ICSI and microinjections, and do the biopsy for the embryos. Therefore, if we can use the artificial intelligence to help us to save time, and don't burn our embryologists out, therefore we're going to use it. Even it's going to be expensive. Yes, it's very expensive for labs to switch to the digital IVF lab, but it's worth it because you're going to help your embryologists to focus on what is important for the lab and you need embryologists for. Therefore, I know that some traditional IVF lab, unfortunately, they use their embryologists for hours and hours to do this quality control that they can use the artificial intelligence for them. But I really recommend that they're going to switch to digital lab to save the time, and especially nowadays that we have this technology available to us, why not? We have to use them. And then for our lab, we use the probes for the carbon dioxide, oxygen inside the incubators, and temperature and humidity for the inside the incubators. And for the room, we check the humidity and temperature of the room. And when we freeze the embryos, we put them in a liquid nitrogen tank, and we check the outside and inside temperature of liquid nitrogen tank to just avoid any accidental dropping of the temperature for those tanks. As you can see in this picture here, this is one of the programs that we use to do the quality control, and based on this, we do the quality assurance that will happen monthly, but this is the daily check, and it's going to be a real time. If some of these incubator guys showing here, showing that is not working properly, the temperature, carbon dioxide or oxygen is dropping, then we can just see it in a real time, even sometimes happen in midnight, and then we can just fast go and check that, and then take the embryo out and put it in the other incubator if there is an embryo in that incubator. And these are very, very important for each IVF lab to make sure that the embryo development never going to stop. Okay, the next things that I told you, the time-lapse incubator, that as you can see, this is the one of the chamber in the time-lapse incubator, which is helping the embryology to save time again, and making the lab more digitalized. And the data that you can get from these type of the incubator is going to be very useful and help embryology to choose the patient, to choose the embryo for the patients that they're going to be more helpful to get them pregnant, and one transfer instead of multiple transfers for the patients. We can manage that by this using all of these technologies. Another thing that I recommend for having the efficient digital lab is having the effective patient record keeping, which this electronic medical recording is very important, that you have them very efficient, and you have the computer department that's going to back up all of your data, because we don't have any paperwork to go back and check for the patient data. Therefore, we are just depends on these digital data. And then if something happened in middle of the day working, we need to have access to our computer department, and some person is going to be just help us to make sure that we get the right data, and we can have access. If sometimes we have some glitches if they are here there, then they can just in a real time, they can just take it away. And these are very important things that and details that when you have digital lab, you need to make sure that all of this is from the time that you build this lab and you design this lab, you think about it and make sure that you have some solution for it. Otherwise, it's going to be mess up and it's going to be disaster. Okay, then another thing probably you heard a lot nowadays in all of the just unfortunate news, that there are some mix-up for the patients, that another embryo just transferred to the one patient that it's not belong to that patient. And a lot of things happen, then they transfer, the patient wants a female to transfer to them, and then they transfer male to that patient, and patients are not happy. And these are the things, the lawsuit that going on right now in this unfortunately field. For avoiding that, we recommend to have an e-witness, and we use the e-witness in our center, which we use the radio frequency identification labeling system and barcode. We have 2 digital witness system for all of the steps of embryology lab for one patient. Plus, we have the person witness. These 3 witnesses help us to make sure that never ever that mix-up happen, because that is the catastrophe. And we have to avoid it in any way possible that we can. Therefore, even pay a lot of money to have this e-witness implantation, that IVF lab needs to do it, because this is very, very important and help them in future to just going to avoid these catastrophic events. And that is something that we really make it very important in our lab. And we have 2 e-witness and one person witness to make sure that from the procedure A to procedure Z, that all of these steps going to have a witnesses, and all of them goes past the witness. And this is very important, and I really recommend for all of the fertility center to implement that, and to avoid all of these unfortunate events that right now are happening. Okay, then for this purpose, you need to have your lab director and your technicians, embryologists, to be the tech savvy, and you need to send them to some training classes for softwares, and to make sure that you train them very well, and they have access to the department of other computer department and other people that can help them to solve the problem if they have any digital problems and EMR problems at the time that they work with the patients. Okay, then the last slide, I would like to emphasize that it doesn't matter if you have the traditional IVF lab or you have the modern IVF lab, you have to have a leader. And then leadership in IVF lab is very, very important. And this is going to help the embryology to stay in your lab. And right now, I know that embryology burnout is very, very common, same as a nursing burnout, because we don't have enough embryologists, and especially senior embryologists who work in this field. And embryologists sometimes working between 10 to 12 hours, and this work is mentally makes them tired, and they cannot really go on for 12 hours sometimes. And that is why some Gen Z embryology, they want to change their job, because the stress level is so high, you're working with the cells. And second, the hours are so high and they have a lot of responsibility in the lab. And these are all makes them very stressed out and burnout. And when we have the leader in the lab, it helps them to just have the lab that make the embryologists happy. Therefore, the most asset in our lab, which the embryologists, they're going to be so much happier than the traditional IVF lab. Then this lab director needs to go and take the leadership courses. And this is very, very important. I want to emphasize here. If you run any medical lab, you need to take this very important, and this is necessary, because you cannot just have the lab director and say that, okay, everything's going to be fine. No, that lab director needs to be the leader, need to take these leadership courses. And this is something that I think is going to be more important for the IVF lab and medical lab in future. And we're going to emphasize on that. And second and last thing I want to emphasize is we need to have a mental health evaluation for the lab director and embryologist. Make sure that they are standing in a good, and then if not, help them to go and get help. And these are very important to help your embryologist be happy in your lab, and listen to them and help them to have their job development, which all of the embryologists looking for, and not just working 10 to 12 hours like a technicians, because all of these embryologists are scientists. Okay, I'm going to finish my talk by telling that I emphasize on the leadership. And then if you have any questions, I'm here to answer your question. Thank you so much for your time and attention.
Thank you so much, Dr. Asemi. All right, let's open this up for a couple of questions before we move on to our next presenter. We'll look at the chat here. All right, Dr. Brooks says this was a fascinating and amazing presentation. Thank you. Lisa says incredible detail. Absolutely. So not my wheelhouse. I found it very, very interesting. Thank you.
Thank you so much.
George also says the same thing.
Thank you. I really appreciate it.
Dr. Katie Chang asks, are infertility rates rising?
Yes, unfortunately, yes. And because of lots of preservatives, hidden sugars in our food, and a lot of stress daily, unfortunately, they are in rise. And then sperm concentration and motility in young men are declining, and we have diminished ovarian reserve for the women, unfortunately, in the early age compared to the previous generations. Therefore, all of this going to just make these infertility rates higher than before. And probably we need a lot of help with the public health to make sure that if there is something that is going to makes them infertile, like a preservatives in our food, hidden sugar in our food, these are the very good study that could happen just hand to hand with infertility and PhDs in the academic, and then make sure that what caused this much infertility nowadays in our society.